Welcome back to the course of plant developmental biology. We are still discussing about the molecular genetics approaches for the studying plant development. So, if I recall previous class, we were discussing about the reverse genetics based approaches to study a particular developmental processes in plant, and we have covered how to identify a particular gene, how to choose a particular gene to study its function, then how to analyze its differential as well as temporal and spatial expression pattern at a particular stage of the development or in a particular organ and tissue. Then we have taken this gene for the functional study, which two approaches mainly we have studied. One is the gain of function approach and the loss of function approach. Now, so we have a gene, we have its function, we know what kind of phenotype this gene has when we when we downregulate this gene or when we silence this gene. And second thing, we have a phenotype or we know what happens if we ectopically overexpress this gene. Now, the next part what is remaining in this one is that if you have a gene, what is the next? And next will be the gene interaction study or the mechanism, what is the mode of action, how a particular gene is regulating a particular process. This is important. So, and another thing that one particular developmental process is not usually regulated by a single gene. So, you might end up identifying multiple genes which might having similar phenotype or same phenotype. Then how to study what kind of interaction, what kind of relationship all those genes have for a particular in a particular developmental process. The first thing is that let us assume that if you have two genes and if you have mutant, loss of function mutant of both the genes, and if you find same phenotype, if you see that in both the mutants phenotype is same, then there is a question, is the first thing is that is the both mutants in the same gene or in the different gene. How to rule out this possibility? This you can do by genetically, and you can study the genetic interaction. You can make, if I take one example, so if one mutant which is M1 and another mutant which is M2, both are having same phenotype, both are recessive loss of function mutants, and let us assume the phenotype is short root. If I make a cross and if both the mutations are in the same locus, what is going to happen? You know that the genes exist and there are two loci, and if these mutants are in the same gene, then you can expect that somewhere here is the mutation. Here the site of mutation might be different, but in the same gene. But if you make a cross and go to the F1 generation, what are you going to have? You are going to have one allele from one mutant and another allele from another mutant. But in both the cases, but in this heterozygous F1 plant, both the alleles are disrupted. So, you are going to have F1 plants with mutant phenotype. If this is the case, then you can say that these both independent mutants, there the mutation site is in the same gene, probably in the same gene. On the other hand, if in this mutant mutation is, let us assume, in gene A, and there is another gene B which is also playing role in the same developmental process, and in second mutant, if mutation is in gene B, but both are homozygous, when you cross them and go to the F1 generation, you are going to have gene A disrupted from here, but you will have allele of A as a normal from another plant. Here you will have gene B allele normal from this plant, but here you are going to have B mutated. But in this heterozygotes, what will happen that the wild type copy, both the wild type copies of the gene will complement the mutant phenotype, which means that in F1 you are not going to get any phenotype. So, this kind of genetic analysis you perform, then you can tell that whether both the mutants are in the same gene or in the different gene. Then another kind of analysis. So, basically here what we are trying to discuss today is that what are the different types of interactions. The first interaction we can call is the genetic interaction. If genetically two mutants or two genes are interacting, What does it mean? It means that if there are two mutants or two genes which are regulating same developmental pathway, let's take example, if both are regulating root length, then are they working in the same pathway, are they working in the different pathways? This we can solve by having genetic analysis, by performing genetic analysis. How you can do? You can make a kind of combinatorial mutants. You can combine both the mutant and look the phenotype. We'll see the example. Another things that you can identify suppressors or enhancers of the mutants. So you take a mutant and then again mutagenize and try to find another double mutants where either the previous mutant phenotype is enhanced or suppressed, and then you establish their interaction through the genetic analysis. So this kind of interactions are called genetic interactions. Then second thing is that if they are genetic interacting, or they may be interacting, may not be interacting, the next question is come, are they physically also interacting? So there are lot of transcription factor, lot of developmental regulators, they are known to interact directly, protein-protein interaction, and they can make a higher order complex, and that complex is actually regulating the process. So how to study protein-protein interaction? That would be another thing, and to establish if you have more than one regulator. Second thing is, this is particularly protein DNA interaction or RNA interaction. This depends. Let's assume that if your developmental regulator is a transcription factor, which means that it will go and bind to some DNA element to activate gene expression or for its function. So to establish, so there could be two kinds of genes. One genes which your transcription factor is directly regulating, it is going physically binding to the promoter or the cis regulatory elements of your gene and activating or repressing the expression depending on its nature, or it is indirectly regulating. In indirect case, your transcription factor X is regulating gene A, and then gene A is regulating gene B. So if you take X with respect to X, B is also regulated by X, but X is not directly regulating B, it is indirectly regulating B. And then you can, based on this kind of genetic regulatory physical interaction, you can build and you can understand what is the regulatory network formation. You can also take help of the co-expression analysis. So if you check a particular tissue or particular developmental stage and identify what are the genes which are co-expressed, you can basically predict that there might be interaction between them if both are present in the same cell or in the same tissues. So we'll take one by one. So first start with the genetic interaction. So genetic interactions, so as I said that you can do by identifying either modifiers, which could be either suppressor or enhancers. One possibility is that if you have a mutation, let's assume you have a mutant where mutation is in this particular gene, but if you take and try to identify a suppressor mutant, if you mutagenize, there is again possibility that another mutants which happens in the same gene, either at the same site or at the different site, and the second mutants might restore the function of this particular gene. Such kind of mutants are called intragenic suppressor. In another possibility is that you have a mutant, you have a phenotype, now second mutation you identify in a totally independent or different gene, and if this mutation is suppressing phenotype of these mutants, then it is called extragenic. So you can identify extragenic mutation, you can establish their genetic interaction, you can establish their physical interaction, you can establish their regulatory interaction and try to understand what is the mode of action. When we are analyzing whether they are working in the same pathway or different pathway, if they are working in the different pathway, then it means that there are more than one parallel pathways going on, and both the parallel pathways, they are regulating the same biological process. But if they are working in the same pathway, then you have to establish which gene is upstream, which gene is downstream. Is A regulating B or B regulating A? So which gene is upstream and which gene is downstream, and how you can establish this, again through the genetic interaction. And the common way of doing it, that you make a different mutants, higher order mutants, combinatorial mutants, double mutant, triple mutant, and then analyze. Genetic redundancy we will take separately how to study this. So I will take some of the example. This is a well studied case of a floral transition. So basically what happens in the plant, up to a certain time point, plant undergo the process of vegetative growth, then it decide to transition. But the transition from vegetative phase to reproductive phase is a very important process in the plant to ensure proper sexual reproduction or successful sexual reproduction. Therefore, it is regulated by multiple regulators, multiple pathways. And but these pathways, some of them are working in independent manner, and then there are interdependent manner. There is a huge amount of crosstalk, but how this crosstalk was established? For example, if you look here, there are 4 pathways: photoperiod, autonomous, vernalization, and gibberellic acid pathway. And all these pathways, this is a very simple picture, but they are these pathways are quite complex, and all they are eventually regulating the transition, floral transition. So, I will take few of the example, and if you look, so if you look the photoperiod means the amount of light a plant is getting. Based on that, we can define entire plant into 3 category, either they are long day plant, short day plant, or day neutral plant. So, in long day condition, what happens, one gene which is getting activated is constants, and then constants activates 2 pathway, one is FT and SOC1, and FT and SOC1, they are having their independent or parallel pathways to regulate the floral transition. How to establish this? If you look this mutant phenotype, so here the total number of leaves is counted when there is a transition occurs. If you have more leaves, then it is delayed flowering. If you have less leaf, then it is early flowering. So, if you look at typical wild type plant, so wild type plants, they have usually approximately 15 leaves before they make 15 rosette leaves before they transit from vegetative phase to reproductive phase. But if you look these FT mutants, they are making almost 40 leaves before the transition, which means that there is a huge delay in the flowering. But if you overexpress constants, if you increase the amount of constants, you can see that there is early flowering. So, even less than 10 leaves are made, and plant has already undergone the process of flowering. But another interesting thing what you see, so this tells that FT is activator of both are activators of the flowering. But what happens if you take constant overexpression in the FT mutant background? The constant cannot induce the flowering, which means that constant is important in activating the flowering, but it is working through FT. So, if you do not have FT, constant is not able to activate the flowering. So, it means that there is an interaction, constant is upstream of FT, and FT it is regulating through the FT. Similar kind of things if you look here. Another genetic interaction you can establish from here. So, again this is wild type. If you have wild type, you have around 15 leaf at the time of flowering, but FT mutants, it is having delayed flowering. Another gene which is called SOC1, SOC1 is also delayed flowering, but if you combine FT and SOC1, the phenotype, the plant is further showing further delayed flowering, which means that this kind of phenotype is called additive phenotype. So, both the mutants are showing additive phenotype. This tells that they are working in 2 independent pathway. So, FT is working through this pathway, SOC1 is working through this pathway. If you only mutate FT, there is a effect. If you only mutate SOC1, there is a effect, but if you mutate both the parallel pathway, the effect is enhanced. Similar kind of genetic analysis has been done here to show that agamus like 17, another gene which is even working through a different pathway, but this is also downstream of constants. As you can see in the constant mutant background, the expression of both FT as well as AZL17 is decreased. So, constant is activating AZL17, and AZL17 is also activating finally AP1 and LIFI. So, the final point of regulation is quite conserved, but the upstream pathways are different, quite different. So, this kind of genetic analysis will basically allow you to place all these mutants or all these genes in a particular pathway, and to generate a kind of genetic pathways. Another important thing is the genetic redundancy. Genetic redundancy happens that sometime if there is a family of a genes, there are more than one members in a particular family. What happens that they all are some of them are doing the same function, which means that if you have mutant of one gene, you might not see a significant effect. But if you mutate more than one genes, you start seeing the effect. This is called genetic redundancy. So, for example, if you look this is wild type plant and this is plethora. Plethora are AP2 domain containing transcription factor, very important class of transcription factor which regulates meristem function, stem cell maintenance as well as flowering. But here I am showing the effect in the root apical meristem. If you mutate plethora 1, these are two different mutant of plethora 1, you do not see a significant defect in the root growth. Similarly, when you mutate plethora 2 alone, you do not see a significant difference. It is quite similar to the wild type. But when you make a double mutant, when you mutate both plethora 1 as well as plethora 2, you can see that root growth is very strongly inhibited. They are very short rooted, which means that both plethora 1 and plethora 2, they are working in a genetically redundant manner to regulate the root growth, and single mutant of them do not have a significant effect. Similar kind of things you can look here. Here the effect is on the lateral root. So, primary root development is normal, and these are ARFs. ARFs are auxin response factor. They are also a class of transcription factor, but they works in the auxin mediated signaling pathway. So, for example, I'll focus on two of auxin response factor. This is ARF19, this is ARF7, and this is wild type. So, if you compare with the wild type, ARF19 and ARF7 alone, they do not have much effect on the lateral root development. So, you can see that primary roots are developed, and these primary roots has a significant number of lateral roots. But when you combine both, when you make a double mutant where ARF7 as well as ARF19 both are disrupted, you can see that primary root is very well grown. It is equivalent to wild type, but this does not have any lateral roots. So, this also suggests that auxin response factor 7 and 19, they are genetically redundant in regulating lateral root formation in Arabidopsis. So, this kind of genetic analysis will allow you to establish a genetic relationship between or among various regulators of a particular developmental pathway. The next thing is that if they are genetically interacting, what would be the next question? Are they physically interacting as well? If this is so, how to study them? There are many ways to study protein-protein interaction. Some of them is highlighted here. This is IS2 hybrid, this is affinity purification based mass spectroscopy, and this is bimolecular fluorescent complementation. These are few very commonly used. So, in IS2 hybrid, what we are doing, you take a gene and you basically so both the genes. So, here what is important that you have to have both the gene cloned in a different vector. One will be fused with the bait and one with the prey. And if both the genes are basically interacting, they will interact, and then the bait and prey, they will come together and they will give some kind of signals. In affinity purification mass spectroscopy, here basically based on the affinity column, or you can have a kind of pull down, and then you pull down some proteins, and along with that proteins there will be lot of proteins are coming, and then identify through the mass spectroscopy, establish their identity to know that which genes are getting interacting. Similarly, this is in principle this is similar to the IS2 hybrid, but here the mode of detection is the fluorescent base. So, if you have a fluorescent protein which has two domains, but if these two domains are together, then and only then they will give the fluorescent. So, you clone them in the separately, use one protein here, another protein here, which you want to see whether they are interacting or not. If these proteins are interacting, then they will bring both the domains together. When A and B domains are together, they will give a kind of fluorescent signal. Then you can basically show whether they are interacting or not. So, here are few examples of this protein-protein interaction. For example, if you look AP1 and Sepleta3 AP3PI, you will study in the next class. They are very important genes which are regulating floral organ development in Arabidopsis. And SUU, SUU, this is another gene which is important, and here what they have tried to check that whether SUU is interacting with these ABC genes or not, and this has been done by the IS2 hybrid. And what you can see, this color signal will tell there is an interaction. If there is no color, then there is no interaction. So, if you look this assay, you can clearly find that SUU is interacting only with AP1 and Sepleta3, but it is not interacting with AP3, it is not directly or physically interacting with PI. Then if you have identified AP1 and SEP3 are interacting with C1, you can also establish through IS2 hybrid that what is the domain of interaction. So, this AP1 and SEP3 both are matched domain containing transcription factor. They have one domain which is called MIK domain and another domain is C terminal domain. So, here specifically they have taken only MIK domains of both the factors and C terminal domains of both the factor, and they have checked the interaction. You can clearly see that only C terminal domain are showing interaction, but MIK domain is not showing interaction, which means that you can tell that AP1 and Sepleta3 they are interacting with SUU through their C terminal domain. Okay, another assay, this is done for another two very important transcription factor during adventitious root development in rice. ARF3 is an AP2 domain containing transcription factor, VOX11 is a homeobox domain containing transcription factor, and both are very important for adventitious root development. And here through the pull down what they have done, so they have basically fused ARF3 with GST tag and VOX11 with his tag, and you can pull down using anti-GST antibody or anti-his antibody. And if you are using anti-GST for pull down, and then you can do immunoblotting using anti-his antibody. So, if ARF3 and VOX11 are interacting, then if you pull down ARF3 VOX11, you can detect VOX11. If you pull down VOX11, you can detect ARF3, and this is in vitro way of doing the detection. And here you can see that either you use anti-GST or anti-his, in both the cases you can detect the pattern. Another way of doing protein-protein interaction is co-IP, co-immunoprecipitation. This you can do in vivo. So, you can basically make a transgenic plant where you already have a tagged protein. So, for example, this is flag tag. So, you can tag your protein with any tag, and then you can use antibody against the tag, and you can do the pull down from the total extract from the plant, and then detect or do immunoblotting with the another gene which you want to show the interaction. This is from the same study, and this is your BIFC methods, and here you can see when both the proteins are interacting, you will have LO signal. If they are not interacting, there will not be LO signal. This is example of pull down followed by mass spectroscopy. I will not go in the detail, but why I want to tell here that you can do at the global level. So, you can tag a protein, you can use that tag and antibody against the tag, you can do the pull down, and then you can identify all the protein through mass spectroscopy at the global level to identify all the proteins which are interacting with a particular protein. Then second study which is important is DNA protein interaction. As you know that most of the master regulators of the developments are transcription factor. So, it is very important to identify what are their binding domain, what are the DNA elements where they bind, and how they regulate, what are their direct targets, what are their indirect targets. So, how to do, there are again plenty of techniques available, but few of them I am discussing here. So, there are some in vitro way of doing this is footprinting, electrophoretic mobility shift assay, and then IST1 hybrid in IST1 hybrid you can use a DNA and then identify what are the proteins which are identified to that DNA elements. These two are the more kind of physical assay to study the interaction, FRET and SPR, and then CHIP assay, chromatin immunoprecipitation, which is being very extensively used to study the protein-protein interaction. So, This is one example of electrophoretic mobility shift assay, gel shift assay also this is called. So, in this case what you can do, you can take a putative binding site DNA element, you can make a small probe, and then this probe can be labeled with radioactive nucleotide, and then you mix with your protein. And what happens that if your protein is binding with these elements, and if you run the gel and detect the binding using any mutants, you can see a shift. So, now this is your free probe. If your free probe is bound with the protein, then you will detect a protein or at the higher molecular weight size. Then you can tell that your protein is basically binding to this element. To confirm it, you can mutate your binding site, putative binding site, and show that when you are mutating this, the binding is totally disappearing. As you can see here, there is no binding, but here there is binding. Okay. Then chip assay, as I say, this is very important. So, chromatin immunoprecipitation, this you can do at the gene level, you can do at the global level, the way you want. Here what you want, basically you have a putative binding sites. So, what you do, you allow your protein to bind to the chromatin. If you have a transcription factor, it will go and bind to its target, then you fix it, you may cross link it. So, your protein will be permanently or covalently bound to the DNA, and then you extract total chromatin, make a small fragment, and use antibody against either your protein directly, or if you have some tag associated with your protein, and pull down those chromatin regions or those fragment of chromatins which are bound with your protein, and then you treat with the proteinase. So, you will remove your protein, now you have a DNA fragment only. So, either you take that DNA fragment, and if you know what is the putative binding site, you can design a specific primer and you can amplify through PCR, QRT PCR, or if, qPCR, or if you do not know the sequence, you take those fragments, clone in some vectors, sequence them, and identify what are the binding sites. So, these are some examples. For example, if you look, these are the putative binding sites of AGL24, which is MATS domain transcription factor, and this is tagged with the HA tag, 6 HA tag. So, if what you can do, you can basically do the chromatin immunoprecipitation using antibody against the HA, you will find the fragment, and you want to see whether AGL24 is binding to SOC1 promoter or not. These are the putative binding sites for the AGL24 in the SOC1 promoter, and then you check by RT-PCR or by PCR, simple genomic DNA PCR, for the different region. And what you can see that this region is showing maximum enrichment, which means that this region is getting enriched when you are doing chromatin immunoprecipitation for 24, which means that binding is very high at this region. Here this is low, here this is low, but if you look this region, the binding is very, very low. Similar kind of study has been done here, where targets are direct targets of MATS1, another transcription factor in rice, has been studied, and here you can see this analysis has been done to show that MATS1 is directly associated with the chromatin of all these direct targets. This is the way to study protein-protein interaction or DNA-protein interaction, and this is very important to basically understand what is the mechanism of a transcription factor. Then you will move more to understand the regulatory interactions among the regulators and their the pathways which are being regulated by developmental regulators. One way of doing is that meta-analysis. You can do the co-expression analysis, and there are all these data which are already published or submitted to the database. All these database can be retrieved, and lot of expression analysis, and actually there are lot of database has been generated where you can you just put ID of your gene, you can find what are the genes which are co-expressed with that gene. If they are co-expressed with that gene, you may expect that there must be some interaction, there may be some interactions among themselves, and this kind of study has been used to identify. For example, if you look here, a global co-expression network approach for connecting genes to specialized metabolic pathway. So, if you combine data, if you look the co-expression, then you can basically identify those genes which might be responsible or which might be functioning in a similar developmental pathway or similar metabolic pathway. Then another very important thing, particularly with transcription factor, is if you want to nail down that what is the mechanism, how a particular transcription factor is regulating a particular developmental pathway, you have to identify what are the genes which are regulated by this transcription factor. And this you can do again as a gene level, or you can do at the global level. For identifying this kind of genes, the global techniques like microarray RNA sequencing can be used. If you quantify total expression of the genes or global expression of the genes in the wild type and mutant of a particular transcription factor, you can identify what are the genes whose expression levels are altered in the mutants, and then you can establish that they are basically getting regulated. And then this kind of study has been taken very recently to identify global genes regulated by plethora genes. So, as I said, there are multiple plethora 1, 2, 3, 4, 5, 6, 7, and then what it has been done. So, plethora's genes was fused with the glucocorticoid receptor. If you recall one of the previous lecture, there I have discussed what is glucocorticoid receptor. This way you can control activity of your transcription factor by regulating its translocation from cytoplasm to nucleus, and then when you induce and after induction you can perform microarray and identify what are the genes which are getting induced or what are the genes which are getting repressed. So, by doing this, they have basically identified the genes which are shared by 2 plethora, 3 plethoras, 4 plethoras, 5 plethora and all 6 plethoras, and they have also identified what are the genes which are specifically or uniquely regulated by either of the plethora, either activated or represses. But through this analysis, through this global analysis for all this, there was a very interesting observation here that all the activated genes, if you look here, so they are expressed in the meristematic genes. So, which means that in the meristematic genes or in the meristem domains, the genes are mostly getting activated, whereas repressed genes are mostly expressed in the elongation domain. So, by doing this kind of expression or genetic network analysis, you can predict genetic regulation. Similar kind of things has been done here, where architecture of gene regulatory networks controlling flower development in Arabidopsis thaliana. So, as I said that the flowering starts from the transition from vegetative phase to reproductive phase, and then once this transition has occurred, the floral primordia under the floral meristem undergoes the different developmental stage, and targets of different developmental stages or transcription factors at different developmental stages has been already identified in different stages. And then if you can take all this data and if you can do the co-expression analysis, basically you can establish a regulatory relationship among the regulators, and you can identify the genes which are commonly regulated or the genes which are specifically regulated by a particular regulators. Okay, so this will be probably last of today's slide. So, here what I am trying to tell you that if you want to identify what is the direct downstream target of a transcription factor, this could be one very good strategy which has been very successfully used with one transcription factor, which is MATS1. So, if you remember my previous class, I have already told that MATS1 is a very important regulator of floral organ development or flower development in rice, and if you mutate MATS1, basically you will not have a proper flower development and these plants are sterile, it will not make seeds. Then in that case, if you want to study this gene, you will have to generate a very special thing. So, here what has been done, that MATS1 artificial microRNA against the MATS1 has been developed, and then at the same time MATS1 delta GR construct was provided. So, basically what will happen, this artificial microRNA will go and silence this is designed in a way that it will not silence the delta GR fusion version of the MATS1, it will only silence the endogenous gene, and then this delta GR MATS1, if you induce with dexamethasone, it will go and complement the phenotype. So, you will have the plant, as you can see, this is wild type. When you do not complement with delta GR, florets are like this, but when we complement with delta GR after dexamethasone induction, flowers are very normal. Then you have to perform a very specific experiment. If you take these lines and if you mock, means if you do not treat with dexamethasone, what will happen, there is a down regulation because MATS1 will be down regulated by this gene, and you can see what happens that these genes, the expression is going up and this gene expression is going down. So, when MATS1 is down regulated, 34 is induced, 55 is down. Let us take the example, which means that MATS1 is negative regulator of 34 and positive regulator of MATS55. But when you induce with dexamethasone, the induced expression or repressed expression of the targets are coming back to the normal. But if you do this in presence of cyclohexamide, so cyclohexamide is a protein synthesis inhibitor. If you do that, then what will happen? It will activate only the direct target, indirect target will not be activated. And if it is direct target, then you will see the effect. If it is not directed target, you will not see the effect. So, what happens here, when you put in presence of cyclohexamide, this is going down. This is the effect, which tells that these genes are directly regulated by MATS1. And this approach has been used in combination with the chip sequencing. So, basically chip sequencing is very similar to the you do chromatin immunoprecipitation, identify those genes and use for the sequencing or chip sequencing. And then through this study, 3 important genes has been identified which are directly regulated by MATS1, and then you can use and identify their regulatory networks. You can establish their regulatory networks. So, this way we will stop here, and in next class we will start next chapter of plant development. Thank you very much.